Sorry, thank you.
Thanks. Thank you. And I, too, would like to thank the organizers for offering me this opportunity to share some of the work that's been ongoing in my group over the last several years. So I first became interested in the potential of mother-to-offspring transmission after hearing reports of young fawns less than 4 months of age that had been culled and were CWD positive. And in those early days, as Ed just described, we were not able to detect prion positivity until animals were at least 6 months of age by the conventional methodologies of IHC and Western blot analysis. And so it led me down a path to start thinking about the potential of another route of transmission, that being mother to offspring. So we began by asking that question, trying to determine whether or not the disease was potentially spread in utero, whether or not it was occurring during parturition, or whether or not milk may be a piece of that puzzle. We also were curious as to whether or not there may be a role for males in transmission of the disease as well. So we've been utilizing native cervid hosts for these studies, and as Ed mentioned, we've also been using the cervidized transgenic mouse for bioassay. So this is a mouse, again, that overexpresses the prion protein, allowing us to get a better understanding of infectivity of particular fluids in body tissues. For these studies, we also realized that white-tailed deer will probably not be the best host for these studies. They breed once a year, whereas the Reeves muntjac deer is a polyestrous deer. It is breeds well in captivity. It's a smaller animal, as you can see here, in comparison to white-tailed deer. Nevertheless, it does not necessarily make them any easier to work with. They're pretty jumpy to work with. Most importantly for these studies, they are susceptible to CWD in about the same time frame as other cervid species. So about 24 months, we start seeing clinical disease in these animals. They have a very similar distribution of the disease throughout their body as well. So in these initial studies, we inoculated dams with CWD by oral routes of inoculation, and then we bred them during either early stage infection or late stage infection. So basically, we just took the 2 years, we divided it in half. Animals that had been infected for less than a year, we termed as early infection. Animals that had been inoculated and were more than a year, we called late infection. We then introduced them to live breeding with intact males, and as you can see, we got 14 offspring from these 12 dams. So they were able to reproduce efficiently, and in fact, some of them were able to have more than one offspring. Whoops, I think I've gone a little too fast. Let me back up here. Crazy, I can see it here, but I can't see it there. There we go. So we had a couple cohorts of animals that were born from these positive dams, some that were born alive. 3 of the 14 were born alive, and we had a very high population of animals that were full term. They were normally developed, but they were non-viable. So they died either during parturition or a day or two after. In fact, we saw a 60% increase in this non-viability in animals born to CWD positive dams in comparison to our negative controls, which had a viability very similar to muntjac deer out in the wild. So we wanted to follow these offspring that were born alive over time, one, to see whether or not we could detect, as Ed was mentioning earlier, by lymphoid biopsy to determine whether we could see positivity in these animals over time. So we took longitudinal collections over time in these animals, and we also wanted to see whether they would actually develop clinical disease. So in fact, we were able to see positivity by conventional immunohistochemistry, the first being positive at 40 days post-birth. These developed clinical disease between 28 and 58 months post-birth as well. All this work done by Amy Knowles and Aaron McNulty at CSU. So this was able to tell us that indeed that there was transmission to these offspring, but we weren't able to extract out the fact that there may have been maternal exposure through saliva or during parturition, and this maybe was actually what transmitted the disease versus there being in utero transmission. So from here, we bred additional positive dams, and we harvested fetal tissues in utero so that we could talk about whether or not those fetal tissues and the pregnancy microenvironment itself contained CWD without the complicating factors of the fact that these fawns had had some contact with their mothers post-birth. The other piece that we wanted to look at this point as well are these non-viable tissues. So during this time frame, it was not very easy to detect very low quantities of prions in tissues or body fluids. As Ed mentioned as well, we now have in vitro assays that we can look at this. But at the time, conventional IHC had proven negative that there was anything at the maternal-fetal interface. So Ed has mentioned the protein cyclic amplification PMCA that we use, developed in Soto's lab, again, providing an opportunity for us to amplify very small quantities of a positive tissue through a combination of sonication or shaking to build up this amyloid form so that we could detect it by conventional Western blot means in looking at positivity. We did in fact find that there were positive tissues, both from the in utero derived tissues as well as in the non-viable offspring, suggesting that there was CWD present in these tissues. In fact, we were able to find several tissues suggesting maternal transmission, and in particular suggesting the fact that there may be vertical transmission. We found positivity in the placenta, so that intimate interface between the mother and the offspring. So as I mentioned, we orally inoculated these animals, and we used anywhere from 0.5 gram to 1 gram of tissue because we wanted to make sure that we had inoculated them. And in nature, as just described, we presume that there are very low exposure rates and potentially multiple exposure rates over time. So we wanted to ask this question in a group of animals that were free-ranging, naturally exposed. So with our collaborations with the National Park Service, with Jenny Powers and Margaret Wild, and postdoctoral fellow in my laboratory, Anka Solario, we were able to gain access to tissues both from dams, from pregnant dams, and from their in utero harvested fetuses to try to get a better understanding of this. So Jenny and cohorts provided us samples blinded so that we had no idea what we were looking at. And a couple things that we were able to determine. First, in Rocky Mountain National Park, the prevalence rate is about 13% for CWD, and this, of course, by immunohistochemistry, which is used as the standard for detection. So the National Park Service was able to find that 3 of 19 of these dams had positivity in their tissue. By using this highly sensitive in vitro assay, we were actually able to find that about 80% of these dams had positivity in their tissue, suggesting a maternal silent carrier state. When we went on then to look at the fetal tissues, at the time we were using PMCA and found that about 63% of the fetuses that were being carried by these dams had positivity in their tissues. So CWD is present in those fetal tissues. So yes, in this case, we were able to determine that these healthy subclinical carriers had CWD transmission or seeding activity in fetal tissues in these offspring. So then we wanted to look a little closer to the pregnancy microenvironment itself and try to get a better understanding of what might be happening here at this interface. We also wanted to try to convert to using RT-Quick. As Ed had mentioned, we'd been modifying this assay as well as the PMCA to be used for CWD. We had to do further modification of both these assays, including additional amplification to be able to detect in this pregnancy microenvironment. So Ed has also gone through this assay as well, again, utilizing positive samples, intercalating the thioflavin T allows us to get a direct readout with fluorescence that we then now convert to these rates that Ed was showing. And what we found in the pregnancy microenvironment itself was that the uterus, the ovary, the placentomes, and amniotic fluid had seeding activity that was statistically significant above that of negative controls of the same tissues and fluids. So what's the biological relevance of this? So we have seeding activity there. How does that actually relate to whether or not there's infectivity within those tissues and those fluids? So again, we come back to the bioassay. This assay has really allowed us in a shorter frame time than 2 years, but still taking sometimes up to 300 days to detect. to be able to look at proof of principle as to whether there's actually infectivity within the amniotic fluid, within this placentome, the uterus itself, or within the allantoic fluid, which is the waste of the fetus. And through bioassay, we were able to show infectivity in the amniotic fluid, in the placentome, and in the uterus, but not within the waste from these fetuses. We still have ongoing assays to determine whether or not we see infectivity within fetal tissues to date. So what are the potential mechanisms? As was mentioned earlier, we know that over time that prions can actually increase throughout the periphery of an infected animal. And so one of the thoughts was that perhaps we were just seeing positive in this environment because the entire deer was positive, including her uterus in that reproductive environment. A second potential is that the fetus through gestation ingests amniotic fluid, and from early gestation to late in gestation, about 500 mils per day of amniotic fluid is ingested by the fetus. And the third potential that we were seeking was whether or not within the placentome itself that there may be infectivity. As mentioned earlier, we know that blood carries infectivity, and is it possible that at the maternal-fetal interface, where there's only about 3 to 6, depending upon species, tissue layers between the maternal blood supply and the fetal blood supply, whether or not there may be transmission by that route. So one of the first things that we looked at to try to differentiate whether or not this was occurring just because the dam was positive and all of our tissues were positive is we went back to our Quick, and we looked at tissues that were from less than half the disease course. So animals that were still in the asymptomatic phase of disease in the earlier phases of disease. So these were healthy subclinical carriers that we looked at these tissues. And what we noticed was that we actually saw positivity in placentomes prior to being able to see positivity in the uterus, suggesting to us that maybe there was something that was happening again at that maternal-fetal interface, that maybe blood may be a component of that. And so we do hypothesize that blood-borne prions may be part of this picture, and that perhaps trophoblast cells, which are a fetal-derived cell, the first cells of a fetus that are developed, may be part of this picture. So here you can see just a cartoon of the maternal side versus the fetal side, and if we focus here on the structure, the placental structure of the deer, these are the trophoblast cells. This is the maternal blood supply, and this is the fetal blood supply. So these trophoblast cells are phagocytic, and they're also highly motile. So we know that they can move from the maternal side, from the fetal side to the maternal side, and then back to the fetal side. We've also been able to capture blood cells within these trophoblast cells, suggesting to us that this may be a potential mechanism of the transfer of prions from mother to offspring. So we're continuing these studies right now. We're looking earlier in gestation. We're also looking during embryogenesis and perhaps within the germline itself, asking the question as to whether or not these trophoblast cells are actually the Trojan horse that may be carrying CWD from mother to offspring. We're also very interested in looking at much earlier time points. We know that every female is born with all the eggs that she'll have her entire life, and these eggs are constructed from cells from the ovary. So is it possible that there's multi-generational transmission from mother to offspring, mother to daughter, for transfer of this disease? We're also interested in looking at the potential of milk transmission. We, to date, have actually inoculated fawns with milk from either positive or negative dams, these all in controlled environments. We've recently euthanized these fawns and are now using Quick to look at the tissues from these fawns as well as milk from these dams to determine whether or not there's positivity there. We're also looking at the potential of sexual transmission and looking at both semen and testicular tissues from intact males. So we know that transmission and disease progression has occurred in offspring born to positive dams. Of course, in those studies, again, we don't know whether that was because of that contact with saliva or the environment, whether that helped with this transmission. But our further studies helped us realize that transmission in utero occurs as early as the first trimester, and it occurs in both early and late infection. We know that this also occurs in free-range natural naturally exposed herds where we see seeding activity of prions in fetal environments. And we now know that there is actually infectivity within the pregnancy microenvironment itself, within the uterus, at the fetal maternal interface, within that placentome, as well as in the amniotic fluid. So in summary, fetuses are exposed or may be exposed to CWD long prior to parturition and prior to exposure by maternal grooming in contaminated environments. So I'll switch gears now and talk just a little bit about the potential of cross-species transmission and just a bit of an overview. One of the questions, of course, asked is, are non-cervid species susceptible to CWD? There are many species that are sympatric with CWD-infected individuals. And we know through natural transmission dynamics that BSC itself has been able to be transmitted both to cats and FSE, feline spongiform encephalopathy, as well as to humans, variant Cruciate-Jakob disease, and other ungulate species. The other piece of the puzzle that we realize as well is that BSC was amplified within cattle prior to that transmission to humans. So meat and bone meal was fed back to cattle over time. We presume that amplified it sufficiently so that consumption of these products, either by any of these species, is what helped and adapted the disease to be able to transmit across the species barrier. We don't know whether that would be the case for CWD. The one thing, of course, with CWD, where we don't have this amplification through meat and bone meal back to cervids, we also know that in individuals that hunt and consumes cervid species, that it's typically just one family or a small community that consumes that animal versus it being spread out. So one wonders whether the load may be similar in that one animal consuming one family or one small group consuming all of a positive animal. So in studies that were done at Colorado State University to try to take a look at this, we inoculated domestic cats with CWD. So we inoculated them with brain material from a CWD infected deer, either through intracranial inoculation or oral inoculation, and then we watched these animals over time to see whether or not they developed disease. We found, in fact, that between 4 and 6 years after those initial inoculations, that 2 of our IC inoculated animals became positive. During this entire time, those that were inoculated by the oral route did not contract disease. So another piece of prion biology that we better understand is that if you are able to adapt a prion from one species to another through subpassage, that it's much easier for the disease to find itself more like its original host in a new host, so it adapts and is more efficiently amplified within that new host species. And so we did a second passage of this positive material. We took the brains from these 2 cats and inoculated another group of cohorts, either intracranially or by peripheral routes, oral, IP, or sub-Q. We also had a couple cats that just lived with the rest of these to try to determine, actually I think these cats lived with these cats for a while, to try to determine whether contact itself, that there was shedding of the prions from these initial inoculums. What we found over time was that, in fact, the other thing that we know about prions is that we also get a shorter incubation period. We did, in fact, see a shorter incubation period, and we saw adaptation in that all 5 of the IC inoculated cats became positive over time. Again, we allowed these peripheral inoculated animals and these contact animals to go out another 3 years after this time point. None of these animals showed clinical disease. One thing that we were able to show from these animals, as I'd mentioned before, is that adaptation piece. So upon the original inoculation of CWD in felids, we saw a very similar deposition of the prion protein, these red dots here, in the neuropil. of these neurons, which was very similar to what was seen in FSC in domestic cats that were actually that contracted the disease naturally. So the other piece that we saw as well, again in that adaptation process, is that when you take this first initial inoculum and inoculate it into another group of cats, you see a more diverse and widespread deposition of the prion protein that looks much more similar to the original cervid inoculum itself. So again, adapting itself into that new species. So as was mentioned earlier, there have been a lot of experimental CWD infection studies suggesting that there are several species out there that are susceptible to CWD by experimental inoculation, both by IC and oral routes. In particular, cattle, sheep, the ferret, most recently pigs, and then 3 different species of rodents, the Peromyscus, and then voles, a couple species of voles as well. So what about the potential of human conversion of CWD in humans? Well, to date, has been mentioned, there are no known cervid to human transmissions. A couple things that we don't know about is what would that look like in humans. We don't know what an asymptomatic carrier state in humans would look like. There was a study done recently, actually there have now been 3 studies, but a second study that was reported on this spring in regard to the potential of bovid to human transmission. They now have determined that in the UK that about 1 in 2,000 individuals in the UK are a subclinical carrier of variant Creutzfeldt-Jakob disease due to their exposure to BSE over time. We really don't have any idea what that may look like in humans, but again, to date, there are no positivities. There have been a couple non-human primate studies. There's a nice review in Waddell that just came out. Different species of monkeys. It was shown that squirrel monkeys are susceptible by IC and oral route. Rhesus macaques, a couple studies on that. This species is much more similar to we humans than the squirrel monkeys, showing sort of a disparity in no, they're not susceptible, and other studies showing that yes, they are susceptible. So that's still pretty much up in the air as to whether or not CWD and the human potential is there. So a couple things that we again know in these non-human primate studies, there was a greater accumulation in the spleen than in the brain, again suggesting asymptomatic carrier state. So those studies that I talked about in regard to BSE in the United Kingdom, they were actually able to show this asymptomatic carrier state using lymphoid tissue. So they used spleen as well as tonsil and appendix tissues for those studies. So the question then is whether or not there's shedding and further transmission in these animals or not. The other piece is that adaptation that I mentioned before, where the species can become, the disease itself can be either amplified to transmit or adapted from one species to another, allowing for a better transmission. So there have been several studies, in vitro studies, that have been continued on. PMCA, Tim Kurt, doing much of this work and showing that there are multiple species that are that you can amplify in vitro assays. In particular, his work looked at Peromyscus mice and voles. Work done by Kristen Davenport using RT-Quick, also looking at the potential as to whether or not human prions can be misfolded. Her work as well, providing some hints that there are potential for CWD and BSE to misfold in human protein. And then a surveillance study that was done by Jeff Christensen, a small surveillance study in which we, much like the UK study, only using considerably smaller numbers of tissues, we were able to procure materials from both an endemic region in Colorado as well as a non-endemic region, so lymphoid tissues from Colorado and California. To date, none of these studies proved to be positive, and so we were not able to find positivity in any of the human tissues that we looked at. So in summaries, it appears that the species barrier is pretty strong. Experimental transmission of CWD certainly has been seen, though, in species that are sympatric with CWD infected cervids, and the non-human primate data still puts us at a question mark as to whether or not that transmission can occur to humans. So I leave you again with why is it important to study prion diseases. We know that prion diseases, the animal prion diseases, are remarkably similar to their human prion counterparts. They're recapitulated in trafficking, dissemination, and even transmission patterns. We know in animal TSCs that saliva, blood, urine, feces, and mother-to-offspring transmission occur. And in the human picture, we know that there are prion deposition within saliva, within urine, and both in reproductive tissues as well as in placental tissues. We know in humans that transfusion transmission has occurred in variant Creutzfeldt-Jakob disease. And we also know that there are products on the market that basically take antler from a cervid species, and they dry it down, lyophilize it, put it into capsule form, and people are actually consuming that. And antler velvet is basically blood from the cervid species itself. So with that, I'd like to thank my laboratory for the work on the maternal and blood transmission that we work on, in particular Amy Knowles, Aaron McNulty, and Anka Solario. The felid transmission work was done while I was still a graduate student in Ed Hoover's lab. Davis Seelig, Amy Knowles, and myself oversaw those studies. So with that, I thank you for your attention.